Hi class. How are you people doing? I hope everybody is doing good and moving along each day, each week. So I think we are almost at this midpoint. So I hope you're liking the course. I hope you're liking the teaching. And if you have any suggestions or comments, do email me or come to the live office hours. Okay. So today we are going to start the chapter on proteomics. And let's start. Proteomics, it's a word that is derived from two words. So let's see. So the word is the chapter or the chapter is known by the word proteomics. Proteomics is made up of two words. One is proteins, and the second word is omics. And why this is happening? Because now the technology is far superior than before. So initially, when researchers, scientists, they used to do research, they used to mainly focus on one protein. But now we have technology and tools where we can look at least more than thousand, close to 1 million, depending upon the goal of experiment, nature of experiment, nature of research, and capabilities. So proteomics, the word refers to studying or analyzing protein, not one, but a system of proteins. It could be an organism. An organism could be a bacteria. It could be a single eukaryotic cells like algae. And it could be, let's say, a breast cancer cell. It could be a pancreatic cells, which releases insulin. So, or a brain cancer or Alzheimer disease. So we can take a cell and to understand what's going on inside that cell, how the cells, these cells who have turned diseased, they're receiving signals, and what are the abnormalities. Because unless we know the abnormalities, we cannot design the treatment. So proteins, the world of protein in any organism or any system or any cell, not one, but complete array of proteins, that is called as proteomics. Now, before I get deeper, I always do to this my class that we should always remember where we started from. So we started from looking at atoms in periodic table, then we looked at how these atoms come together to form molecules, and we then started focusing on biomolecules. After learning about proteins, carbohydrates, lipid, nucleic acids, we went into cellular structure, and then cell division. Then we looked into mitosis, meiosis, their similarities, their differences, and then we looked into how the DNA does its function by replication. How does it make proteins? By transcription and translation. Right? So, and after these translation, we looked into how the information is inherited from one generation to the next generation by studying some of the basic principle of genetics, which were designed and developed by Gregor Mendel. Now we are going into more applied version of biology, and today we are going into proteomics. So proteomics again is is a study or is a field where a research or a topic or a subject, it focuses on array of proteins, how to decide the function. Okay, so so let's say take one example. So let's say now you know there are many trials for COVID vaccine is underway. There are some encouraging results at this point of time from phase 1 and phase 2 trials. Now scientists and companies, they're going into phase 3 trials. So this is just a diagram to show you how proteomics is important. So let's say there's a protein A in COVID virus, and that could be instrumental in generating antibodies. So it could be a potential vaccine candidate. So is it powerful? Is it effective? So in this system, we can use two mouse. One, a control mouse, and so a control mouse, and then a disease mouse that has infection, right? Or that could be infected, after giving the vaccine. So let's say this is a control where there is no infection. The vaccine is given as a control, whether the vaccine has any toxicity. And the second, this mice could be vaccinated and then it could be infected with the virus, or vice versa. Regardless, what scientists will do, they can look at, because COVID, here it shows drug, but I'm giving you more relevant example in today's date, that is vaccine. So scientists can look at the lung and compare the lung tissue, whether the disease mouse came back or there was no infection prior to giving vaccine. So you can compare, and one can do proteomic analysis to look at what are the molecules different between the normal control mice and the disease mice and the treated mice, right? And they can compare what are those proteins that are impacted by COVID-19 infection or coronavirus infection. And once they learn about those proteins, so they can understand that those proteins play an important role in preventing the infection, because the vaccines, they are triggering those proteins. So information about protein is very important. And that's one of the goal of this course, that you not only learn basics about atoms, molecules, and bonding, but you look at the current research and you can get an idea, and you could be motivated to do higher research, go for research career, go for PhD, or go for public health planning, or become a doctor, physician, many avenues. So that's the nature of this course, to give you basic information and then take you to advanced level. So proteomics could also be, so you know that proteins, they are folded in three-dimensional space. So I won't go in details of this slide, but a protein has a three-dimensional structure. And scientists could study this three-dimensional structure to design more effective molecule. And there are various technologies. So here, so what is protein? Protein is essentially a chemical molecule, and it has a three-dimensional structure. So how this could be manipulated, this three-dimensional structure, to develop more important drugs or molecule or therapeutic design, right? So the take-home message is that unless we know the details of a protein against which we want to make the drug or medicine, we will always be unsure that we have the best medicine in our hand, best potential medicine in our hand. Okay, so proteomics is a research that is focused on array of proteins to understand biological process, or to understand disease processes, disease pathogenesis, and then use the knowledge to devise molecules that could have therapeutic applications. And you can see this slide where you have this is a three-dimensional structure of a protein, and this you see here this molecule here. So this molecule, chemical molecule, is fitting into this pocket of this protein. So unless, let's say this is a protein of COVID called as RNA-dependent RNA polymerase, which helps the the coronavirus to replicate in our cells. So again, if this is a protein that helps the coronavirus to replicate in our lung cells, if we dock a compound that inhibit the activity of RNA-dependent RNA polymerase, which helps in coronavirus replication, so we have a potential drug. So unless we know the structure of the protein, unless we know the atomic detail or molecular detail, we cannot make an effective therapeutic design. Similarly, if you see here in polio vaccines, polio was a major threat to human society in early days. But now, I mean, early days means 40, 50 years back, but now it is a very effectively controlled unless there are some drug resistant polio virus or vaccine resistant. But again, to understand the polio virus, to understand the protein which can elicit immune response, generate neutralizing antibodies. So these days you must be getting a lot of information about antibodies. If you're antibody positive, then you're infected, then you're protected. So what are these antibodies? These antibodies are essentially protein molecules. So to see whether it is to design a neutralizing antibody, because that can combat the virus inside our body, we need to know the atomic detail, the details about this protein, right? So another example is, if you see this Herceptin, this is a drug, this is an antibody that is used to treat one of the type of breast cancer. So cancer is very, very heterogeneous disease. It is not simple, but Herceptin is used to treat one of the type of breast cancer. So unless we know the details about antibody, the breast cancer proteins, we cannot make these antibodies. And this antibody has proved to improve the quality of life of many breast cancer patients. Similarly, this Fabrizyme. So this Fabrizyme is sold by a company in Boston. And this Fabrizyme will help kids who don't have enzyme to digest fat. So if a kid cannot digest fat, then there will not be normal growth. So they have to be supplemented by some enzyme such that their fat get digested, their metabolism is normal. So a company in Boston, they developed this Fabry enzyme. The enzyme called as Fabrizyme. The disease was Fabry's disease, so and so forth. So unless we know the detail about proteins, we cannot understand the disease process. So this chapter is dedicated to make your basic understanding of proteins very, very clear. Okay. So as you know, proteins are made up of 20 amino acids, and the typical structure of protein is that it has a central carbon atom, which is called as C alpha. This is connected to two functional groups, COOH, carboxylic group, and NH2, amine group. So two functional groups. The third bonded is hydrogen atom. And fourth is most important, called as R group. This R group is different for different amino acids. So there are 20 amino acids, and this R group will be different for each amino acid. So what happens in ribosome? Ribosome, there is an RNA, mRNA, and mRNA has triplet codons, and then corresponding to each triplet codon, the tRNA will bring amino acid from within the cells, and then the protein will be synthesized. So how two proteins are connected? The two proteins are connected by, or let's say, to be more specific, two amino acids are getting connected on the ribosome by peptide bond, right? And so what is a peptide bond? Peptide bond is a bond that connects two amino acids, right? So what you see here, so what happened? Let me go back and tell you again. So look here, this is carboxylic group. This carboxylic group reacts with amine group, and when they react, they form a, when these two react, then they form a covalent bond by releasing a water molecule, and then this bond is formed. This bond, CONH, this is called as peptide bond. This is covalent in nature. So if there's a protein which is assembling on a surface of ribosome, then tRNA is bringing amino acids one by one, depending upon the template of mRNA, and then two amino acids are getting connected by covalent bond, and that bond is called as peptide bond. So I just want to quickly show you that there are 20 amino acids, right? If you count, there are 20 amino acids, and what you see in this colored boxes, these are the R group, the side chains, which is different in every amino acid. So you look at any amino acid, they differ from each other in terms of side chain. Rest, everything is same: carboxylic group, amine group, and hydrogen atom that is bonded to the central carbon atom. Okay. So to study, so protein is a world. It is an array of proteins. In a cell, there could be many, many proteins. So it is very important that we classify the proteins. So why do we want to classify? Because we want to have a clear understanding. So proteins can be divided into four types: primary structure, secondary structure, tertiary structure, and quaternary structure. So protein can be studied or can be analyzed under these 4 points, right? So I would say primary, secondary, tertiary, and quaternary structure. Now I just want to cover something very, very basic. And so you know that in English literature, there are 26 alphabets or letters. And these 26 letters, when we talk, we use, so these 20 can form a word, and a series of words can make a sentence. But what is critical here is that these words should have meaning. So let's say now I'm teaching, and if I use word or a language that you don't understand, so there will be a gap of the flow of knowledge. So 26 letters converts into meaningful words, right? And different words have different alphabets. And then words, they form sentences. So there is different, a word will have different number of alphabet. They sound differently, they read differently, right? And they can be put together to make sentences, and then sentences forms paragraphs, and then many paragraphs could come together to form a story, chapter, or let's say your term paper. So what do you see here? So what you're seeing here is that there is a hierarchy here. If we follow, if we know the alphabets, we can make sensible words, sensible sentences, and then we communicate our sense using paragraph, and the complete structure is story. So there is a structure here. Similarly, proteins are made up of 20 amino acids, right? 20 amino acids, which differ in their side chains, and a protein could be made up of, let's say, 10 amino acids to 2500 amino acid. 2500 amino acid. And these proteins, they differ in function. For example, the proteins that are involved in antibodies are different from proteins which are in insulin hormone. And the amino acids which are part of insulin hormone is not the same for digestive enzyme. For example, pepsin is an enzyme that digests our proteins in our stomach. But a different enzyme in the small intestine called as trypsin, it also digests protein, but it's a different protein. So proteins, they differ in their function, in their structure, just like word. So what does primary structure refers to? So primary structure refers to the sequence of amino acids. So for example, if you take an analogy, the sequence of alphabet in a word would refer to primary structure. What is then secondary structure? What is secondary structure? Secondary structure refers to, remember, proteins at the end, they are chemical molecule. And these molecules, they occupy different orientation in three-dimensional space. So they all have a shape. So for example, in this class, you just need to know one shape, alpha helix. Proteins could form alpha helix or beta sheet, right? So a protein could be either alpha helix or beta sheet or mix of that. But there are more components to this that we don't need to know in this class. Then tertiary structure. Tertiary structure refers to when a protein binds to a metal. For example, hemoglobin. It binds to a metal called as iron. Right? So remember, when we feel weak, sometimes a physician speculates that we could be having low hemoglobin. And if somebody is having low hemoglobin, sometimes physicians, they try to supplement the diet with iron. So why? Because iron helps stabilizes hemoglobin. So proteins could bind to iron, it could bind to calcium, it could bind to zinc. So proteins that binds to metal, those proteins have been shown to call as having tertiary structure. Now, quaternary structure. Quaternary structure refers to when proteins exist in when protein exists as a multiple subunit. So again, the best example to give here is hemoglobin. So hemoglobin is made up of 4 different subunits. That's it, 4 subunits. Right? Out of these 4 subunits, 2 are alpha, and 2 are beta. So hemoglobin cannot work as an individual, but it tries to form a team, right? And what is a team here? The team here is that it is made up of 4 subunits, 2 alpha and 2 beta. So these are 4, right? So these are called as tetramer. So a protein could be by itself, monomer. It could be with another partner. So it becomes dimer, or it could be in 3 or 4 trimer or tetramers. So a protein could be studied or classified under 4 structures: primary, secondary, tertiary, quaternary. Okay. So this is primary structure that refers to the sequence of amino acid. Here you see the sequence of amino acids starting from here: asparagine, glycine, phenylalanine, glutamic acid, glutamine, alanine, arginine. So these are the sequence of amino acid. Then, as I mentioned to you, proteins are made up of amino acids. Amino acids are made up of atoms. These atoms, they orient themselves in 3-dimensional state and they form a structure. So one of the structure that you see here is called as alpha helix. And this alpha helix is essentially ribbon shaped. So this alpha helix is ribbon shaped. Now what I want you to pay attention here, so look here, you see here COH interacting with NH, so CO comes from carboxylic acid of amino acid, and that hydrogen bonds with NH group, amine group. So these hydrogen bonds, they stabilize the proteins. So this is alpha helical proteins. Right? Sometimes some proteins are combination of alpha helix and beta sheet. So remember, the take home message is that proteins can form alpha helix, and this alpha helix is stabilized by the formation of hydrogen bond. So let me show you how an alpha helix could look like. So an alpha helix is something like this. This is an helix. And how these helix are stabilized? These helix are stabilized by, let's say, a hydrogen bond. They're stabilized by hydrogen bond. And how these hydrogen bonds are formed? These hydrogen bonds are formed between CO from amino acid carboxylic group and NH from amino group, preceding and following amino acids. So this is secondary structure. So as I mentioned to you, that hemoglobin or a protein could bind to iron. Right? In case of hemoglobin, the beta chain binds to iron. Right? So this is a tertiary structure. Not all protein binds to metal ions, but some, only those protein binds that need metal ion for their function. So hemoglobin needs metal ion. This is an example of quaternary and hemoglobin. Hemoglobin has 2 alpha chain and 2 beta chain, and they're held together because of hydrogen bonding. So not all protein exists as trimer or dimer or made up of multiple subunits or having 4 subunits, but protein that has are shown to be called as quaternary structures. Right? So this is the summary of what I taught here: primary, secondary, tertiary, and quaternary. Now, one thing that I would like to highlight here is that our cells are dependent on protein in a huge way. So cells, because proteins are the major workforce. Proteins are the major workforce. For example, hemoglobin. Right? Although it's not inside the cell, it flows with in our blood vessels. Hemoglobin is part of our red blood cells, which flows in our blood, and blood is a type of solution which has water that facilitates the flow. So nevertheless, proteins are the major workflow. But proteins, after some time, they lose their function, or they begin to lose their functions. So protein have to be replaced, right? So proteins have to be replaced because they start losing their function. So scientifically, we say this term as protein renewal or protein turnover. So new proteins are being made, and the old proteins which cannot function, because if the proteins cannot function and it's still there in the system, then the system will not function with proteins which are not working efficiently. And again, it's a room for starting disease. So like you studied replication system, transcription system, translation system. So likewise, there is a system for protein turnover. And that system is called as proteosomal machinery. Proteosomal machinery is that machinery that helps the protein to. So what happens? Let's say there is a protein which cannot function properly because it has done its role. So what will happen? This protein will be flagged. And then this protein will undergo biochemical changes. And then this protein will be removed from the system. This protein will be removed from the system, and the system that will remove this protein is called as proteosomal machinery. So that is important. Now, if this machinery fails, right? If this machinery fails, then there is a serious outcome. What are those outcomes? So some of the diseases that can occur because the proteosomal system is not working is Parkinson's disease, neurodegenerative diseases, bovine spongiform encephalopathy, scrapie, kuru. So these are the list of some of the diseases that can occur because of the failure of proteosomal machinery. So with this, when we finish the chapter on proteomics.